Welcome to the ImmunoVerse, a podcast that brings the ever-expanding universe of immunotherapy to life through the voices of those advancing this groundbreaking field. I'm Dr. Patrick Hu, President and CEO of Moffitt Cancer Center and a career immunologist. In each episode, I sit down with pioneering experts who have shaped the past, present, and future of immunotherapy, uncovering breakthroughs, challenges, and the science driving this life-saving innovation. Today we have with us Dr. Mario Sznol, Professor of Medicine at Yale School of Medicine and co-director of the Cancer Immunology Program at Yale Cancer Center. With a career spanning decades in immuno-oncology, Dr. Sznol has led numerous clinical trials and been at the forefront of designing combination strategies that enhance anti-tumor immunity. He is especially known for his work in melanoma, renal cell carcinoma, and early translational efforts to understand immune-related toxicities. His clinical insight, coupled with a deep understanding of trial design and patient care, has made him one of the field's most trusted voices. Welcome to the ImmunoVerse, Dr. Sznol.
Thank you, Dr. Hu. It's always a pleasure to discuss science and clinical trials with you. I'm looking forward to this.
So tell us, how did you get involved and interested in immunotherapy?
It's a very long story. It started when I was at Mount Sinai as a fellow. At that time, Dr. James Holland was the director of the cancer center there, or Department of Neoplastic Diseases. He was treating a patient with high-dose interleukin-2, my first experience with high-dose interleukin-2. And I just became interested in it, and I went to the NCI to CTEP as a sort of a drug monitor for immune therapies. And so I became involved with the very first development efforts in interleukin-2.
Yeah, so explain that to people. You were at the NCI working with CTEP. Explain what CTEP was and how that was critical function in those days. I really think the NCI kept immunotherapy alive while no one believed in it, and it wouldn't be around today if it wasn't for those early efforts at the NCI.
CTEP was the part of the extramural arm of the NCI, meaning that it was involved in doing drug development, in providing drugs for drug development in cancer centers, but outside of the NCI. Also supported INDs for drug studies even within the NCI at that time. So think of it as a drug company, the part of the drug company that does the clinical drug development, but sort of at the NCI. And as you know, in those days, most of the immunology, most of the discoveries in immunology, and most of the driving forces in clinical development of immunology, immunotherapy, was actually at the NCI. Think about all the things that Dr. Steven Rosenberg was doing, and of course you came from there, so you know that.
Yeah, so I just remember being really relying on your group back then to run our clinical trials at the National Cancer Institute. You really helped us monitor the trials and get the trials off the ground. So I think it was a critical role. You were there with the early days of cytokine therapy. So explain to everybody what cytokines are and interleukin-2 and how that works and the early approvals.
Cytokines are just hormones that are used by immune cells to communicate with each other. And there are many different cytokines, and their effect is really context dependent. It depends on the state of differentiation of the cell, the type of cell. But in a sense, they drive activation, proliferation, and function of those immune cells. And immune cells can't function without those cytokines. We use checkpoint inhibitors a lot. That's a different way of activating cells. But even when you use checkpoint inhibitors, cytokines are produced that drive the function, proliferation, and activation of those cells. Interleukin-2 was a breakthrough. The initial drug was interferon, but interleukin-2 came next. And interleukin-2 by itself, this is way back in 1985, was producing long-term durable remissions in about 10% of patients with metastatic melanoma and kidney cancer. Actually, it was the proof of concept that immunotherapy could work. And the high-dose regimens were developed within the intramural branch of the NCI by Dr. Steven Rosenberg, and the results were amazing. There are people that are alive today that were treated in 1985 and 86 that received high-dose interleukin-2. If it would not have been for the development of those cytokines, I don't think that we would have been able to continue research in this field.
Then you led a lot of the trials with immune checkpoint blockade, and especially one that became commonly used, and you and Jed Walczak led a trial of Ipi-Nivo anti-CTLA-4 and anti-PD-1 combination. Tell us how that came about and what the results of that combination was.
So when I was at the NCI, I saw Jim Allison's initial publication on anti-CTLA-4. And I became very excited by it. And I knew I wanted to be involved with those drugs whenever they came into the clinic. And I was also very familiar with the work of Li-Ping Chen, who was working on anti-PD-L1 in the clinic. And those drugs looked amazing to me, and I really wanted to be involved. I spent a little bit of time in industry between the NCI and going to Yale, but when I got to Yale, I wanted to make that a focus of my research. So we became involved with the first multi-dose trial of anti-PD-1 and accrued a lot of patients on that trial and saw amazing activity. We knew that drug was going to change the world. So the logical next step was to combine anti-PD-1 with anti-CTLA-4. Not only because it made sense, but because there was a little bit of preclinical data. But in general, Alan Corman was very instrumental in bringing that to clinical trials. You know, he was at Medarex and then at Bristol after that. So we started the study together with Jed Wolchuk. And again, you know, we already knew that anti-PD-1 was an active drug, but the activity we saw with the combination was remarkable. As you know, as it turns out, it's a little bit better than anti-PD-1 alone. It's not logs better, it's a little bit better. But we were very excited about the activity. And actually, those drugs have made a huge difference in the treatment of cancer. And in my disease, in melanoma, we produce 50% long-term survival with a combination of anti-PD-1 with anti-CTLA-4, or now with anti-PD-1 in combination with relatlimab. It's remarkable. And when you think that in 1995, the long-term survival for melanoma was 5 to 10%. Now, when somebody comes in with diffuse metastatic disease, they have a 50% chance of being alive at 5 years. It's remarkable. It's almost a tenfold improvement. And I recently had a patient who had an LDH of 2,000, a very poor prognostic feature in melanoma. Her liver was full of tumor. I could actually feel it down to the umbilicus. It was enlarged. Within 4 doses of the combination of anti-PD-1 and anti-CTLA-4, liver was normal, LDH was normal. I think she's going to be cured of her disease. It's remarkable.
That's incredible. So it must be very fulfilling for you having the regimen that you were one of the PIs on, now FDA approved, and in so many different kinds of cancer.
Yeah, in our field, we only get a few chances to work with really active drugs. If we work with 1 or 2 active drugs in our career, it's a big accomplishment. I feel so privileged that I was able to be involved in the development of those drugs and to see it in the clinic yourself and to see somebody come in with a life-threatening disease. They would die without those drugs and then survive and be cured and live for years because of those agents. It's just an amazing feeling. And I wish we could have that opportunity more than once or twice in our lives. That's why it's so important to have all of this science, all the development of this science. Without the science, without the basic science, without the translational science, without the clinical trials, we wouldn't have these drugs. And this is the only way we're going to improve outcomes for cancer patients in the future. It's the science that drives this.
That's a great point. Now, with that combination of Ipi and Nivo, you did see some additional toxicities, and so you also got very good at managing these toxicities, as you did previously with high-dose interleukin-2, but learning how to manage the toxicities, and importantly, what the science behind the toxicities were, so you could develop new approaches to managing the toxicities.
To be fair, a lot of the basic work and how to manage the toxicities was actually done with the development of anti-CTLA-4. So there were many people involved with that. Stephen Hody, Jed Wolchuk, a number of other people who, and the folks at Bristol helped develop these drugs that learned how to manage the toxicities of anti-CTLA-4. What we saw with the combination was just an exaggeration, an increase in incidence of those effects. And we worked together with the people at Bristol Myers who were sponsoring the trials to develop methods to essentially treat the adverse effects when they occurred, not to prevent them, but to treat them when they occurred. We still don't understand the biology and the immunobiology of what causes those adverse effects, but we're a lot better at managing them. We can treat the adverse effects with steroids and secondary immunosuppressives. This occasional patient does poorly, but most people we can reverse the side effects, and eventually they return to a normal life. What people are working on now is to try and develop ways to prevent those side effects from occurring. First, to predict which patients are going to develop the severe side effects. So we've been working in those areas to try and develop predictive biomarkers for who will develop toxicity. Once we understand who's at risk for developing toxicity, then there are a number of hypotheses that we can test in the clinic about how to reduce the toxicity without affecting the efficacy of the drugs.
So we've talked about cytokine therapy and immune checkpoint blockade therapy as one of the major investigators throughout the country. You're exposed to a lot of different approaches to stimulate the body's immune response, bispecifics, where we try to bring T cells together with a tumor, TCR engagers, T cell therapy, many different flavors of T cell therapy. What's your thoughts about what the most exciting approaches are, and how will we put all this together to help the most patients going forward?
I think the key to all this is the engineering. The engineering of these molecules is amazing. There are already 2 T cell engagers that are approved. Not every cancer will you be able to treat with immune checkpoint inhibitors. Some patients just don't generate a T cell response that you can reactivate with immune checkpoint inhibitors. So you need other kinds of agents. The T cell engagers actually, we think, are actually taking non-specific T cells and redirecting their effect directly at the tumor site to kill tumor cells. There are already 2 T cell engagers that have been approved in solid tumors. It's already a well-established therapy in hematologic malignancies. Tependifus, which is used to treat uveal melanoma, and tarlatamab, which is used to treat small cell lung cancer. In fact, it's the first major advance in the treatment of small cell lung cancer since I was a fellow, and I was a fellow many, many years ago. So it took a long time to see an advance there, and that's the tip of the iceberg. There are so many T cell engagers that are being developed. So it's engineering the molecules to be better and also finding newer targets to go after, which is also constantly evolving. So I'm very excited about the T cell engagers. I think they're amazing. But we need time, we need the investment, we need to study these things in the clinic, we need to be patient. I think the thing that concerns me the most is that we need patience. Some of these agents won't be incredibly active as single agents, but in combination could produce amazing results.
So we have many different approaches now, from cell therapies that you can engineer cells and give them back, to cytokines, to engagers, which are simply molecules that bring the T cells and the tumor together in the body. So there are a lot of different ways and many different drugs. So almost not enough patients out there, not enough investigators like you out there to study all these incredible molecules. Talk to us a little bit about clinical trial design. Are we going to have to do our trials in a smarter way in order to determine which drugs are best for patients and which sequences and series we should give to patients for the best outcomes?
Yes, I don't know that I have the answer to how that's going to be done, but yes, we have to be much more efficient. Putting a patient on a clinical trial is incredibly expensive, as you know, and so the more expensive it is, the fewer options you have. You have to limit the number of things that you do. And for some of these drugs, we need to try several things before we hit on the right combination, sequence, dose, and schedule. So yes, we need to make the trials more efficient. We need to figure out ways perhaps to reduce regulatory burdens to getting these things into the clinic. We need to put more patients on clinical studies, so more people need to be involved. All of those things are really important. I think there's a lot of promising drugs out there. We have to figure out a way to study them. And we have to be, again, expectations are usually very high. In our field, sometimes it takes a few tries before you see success. If you think about immunotherapy, we tried a lot before we really hit on the big drugs, maybe 20 or 30 years before we hit on the really, really active agents. It could take several years before we figure out the optimal way to use T cell engagers, combine them with cytokines, put cytokines together, develop cell therapies, engineer the cell therapies to have optimal effects, especially in solid tumors. But that science is going on. It's going on. It's incredibly exciting.
Well, we still have, the death rates are coming down because of all these advances. We still have 600,000 deaths a year in the United States, 10 million a year globally. What's your prediction? Do you think immuno-oncology and immunotherapy is going to be able to continue to drive these deaths down across the globe?
Absolutely. I mean, I think the next wave of really active immunotherapy agents are T-cell engagers, together or in combination with other agents. And as the engineering improves, those agents will improve and become more and more active. I think the cell therapies will advance over time, and there's a lot of things that we haven't really mined yet. For example, macrophages play an important role in suppressing immune function in the tumor microenvironment. I think we're just now learning how to modulate the function of those macrophages in order to improve immunotherapy outcomes. There are other cells in the tumor microenvironment, T regulatory cells. Last year, we saw a presentation on CCR8 depleting antibodies with significant activity in 1 or 2 solid tumors. Really exciting data. So there's a lot more to mine out there. I think there was a sense because after anti-PD-1 and anti-CTLA-4, the pace of new approvals, certainly lots of approvals for those drugs in different diseases, but not a lot of approval for new drugs. And so there was a little bit of pessimism in the field, but I'm actually very optimistic that we're going to see lots of new drugs. And over the course of the next 5 to 10 years, if we make the investment, we're going to continue to lower those death rates and see more and more activity in different disease types.
So your point is that while there's still we're still at the tip of the iceberg for activating T cells to kill cancers, there's also many other kinds of immune cells, some positive, some negative, that we can manipulate to enhance cancer-specific responses.
Said much better than I can. Yes, I mean there are many more pathways, cytokines, ligand receptor interactions that cause inhibition in the tumor microenvironment. So cells, multiple pathways, lots of things to explore. But you said it much better than I did.
Well, Mauro, you're also a very humble leader, a national leader in our societies. You were the SITC president for even during COVID. So you really had to work through the pandemic for our national meetings, and you did a great job. So talk to us about your service work with SITC and other groups throughout the country.
Um, Primarily with SITC, I think, and actually the reason why I ran for president was really because you asked me to do that. So I credit you for that. SITC is an amazing organization. It's completely devoted to the study of immunotherapy for cancer. It's run by great people. All of us that are passionate about developing immunotherapy are in that society. It's where we exchange information. We work with regulatory agencies to figure out better ways to get these drugs into clinical trials and approved. I think you said it best when you said it was what were the words that you used?
Well, we're a convener of
Yes.
of regulatory, government, industry, academia. We bring people together.
That was exactly what I was looking for. Yes. And it's been very rewarding. And I think really helps to drive the field forward, not just in a scientific way, but think about things like managing toxicity. The society gets involved in teaching investigators how to manage toxicities of these new agents, and that's very valuable for patient care. So at the end of the day, patients benefit from this society and the work that's done in this society.
So part of the point of this podcast is explaining science to people, also telling people that science is exciting, impactful, inspirational, and also that scientists are fun. So you're a fun guy. You're one of my oldest friends. So I know that you showed a picture at these meetings. You get together with some friends and play tennis, right?
I did. We almost at every one of these meetings, several of us go out and play tennis. We call it immuno-tennis. And we're not the best tennis players in the world, but we have a lot of fun. And let me tell you, for me to play with people like Alan Corman, who helped bring anti-CTLA-4 and anti-PD-1 into the clinic, Tom Gajewski, who's a very well-respected scientist here at the University of Chicago, Nacho Malero, who's worked to identify the 4-1BB pathway. I mean, I actually don't feel like I even belong in this group of such eminent scientists, but we do have fun. And by the way, our field, I think, different than many others, we work together, we collaborate, we don't compete against each other. And I think that's part of the spirit of the Immunotherapy Society and investigators.
Well, you heard it again. Science is fun, and scientists are fun.
Yes.
Well, we want to thank our guest, Dr. Sznol, for joining us today, and thank you for going on this journey with us through the ever-expanding universe of immunotherapy. To hear more episodes of ImmunoVerse, make sure to subscribe on your favorite podcast platforms.